Good morning, everybody. I am Bob Handsaker from the McCarroll Lab, and I'm going to give a primer today on structural variation. It's kind of a big topic for this early in the morning, but we'll see how we can do here. And thanks to all the Cubs fans who got up just to come to this. I appreciate it. So this is a genome, not your genome, but it's a human genome, and yeah, hopefully we won't keep blinking like that. But I want to talk here about sort of early technologies in terms of how we could assess copy number variation. And one of the earliest was you can take metaphase chromosomes and you can stain them, and you get this distinctive banding pattern. And if you look at it under a microscope, you can get good at telling the chromosomes apart. And you can learn things by looking at the chromosomes under the microscope. And I'm wondering if anybody in the audience can tell me anything about this genome, anything unusual or strange about this particular genome. Say it again? Yes, exactly right. There's an extra copy of chromosome 21. You know, I don't actually know the prevalence of Down syndrome, although I probably should. I think it's the most common human trisomy, you know, with, of course, distinctive phenotypes. So here's another genome. And anybody have an idea what the deal is with this particular human genome? Any guesses? Yes, this is a cancer genome. The little white arrows are showing you all sorts of problems with this genome: aneuploidies, deletions, duplications. So this is a genome from a tumor cell, and many kinds of cancer frequently have these kind of large-scale chromosomal aberrations. And then the last example I'm going to show here is a particular example of something called the Philadelphia chromosome. It's discovered a number of years ago. It's a common recurrent translocation, I believe, between chromosome 9 and 22, and it creates a fusion gene, and it's implicated in several types of cancer. So these are some of the early things we were able to learn about gross variation in the structure of the genome, principally from this technique of just being able to stain the chromosomes and things that we could see visually in a microscope. So they have to be pretty large, a significant fraction of a chromosome. And then one of the next major advances that helped us in our understanding of structural variation and copy number variation was the invention of comparative genomic hybridization, which takes advantage of the fact that the DNA hybridizes to itself. So you can take a genome and you can hybridize like a normal genome, and you can hybridize other DNA to it. In this case, DNA from a tumor cell in green and from a control sample in red. And you can overlay these and look whether or not there's an excess of green or red fluorescence, because we've labeled the DNA samples we put in. And doing this, you can get a little bit better resolution on what parts of the DNA are deleted and which parts are duplicated, and even guess at rearrangements. And once this technology was invented, it was sort of systematized and automated by doing it on microarrays, and it led to what we call array CGH. So you do this same sort of hybridization, but you can do it with these arrays. And this became sort of a common technique used in the clinic to diagnose things like developmental disorders and to do karyotypic screening, and as far as I know is still probably the gold standard for that kind of thing, although other technologies are probably catching up at this point. And this allowed us to get at certain chromosomal abnormalities and structural changes at this somewhat smaller resolution than a chromosome, but still fairly large scale. And then in the mid around 2005, 2006, we began to have these GWAS arrays that we were using for SNP genotyping. And the GWAS arrays work by hybridizing the DNA and then using fluorescence to see whether or not a particular SNP has like the A allele or the C allele. But people realized that you could look at that fluorescence and not just look at the A versus C ratio, but you could actually look at the raw intensity of that signal, and you could get information about the abundance of DNA. And from this, you could detect copy number changes. If you actually have more copies of the DNA, you're going to have more total intensity on the SNP array. So several groups developed algorithms to call CNVs, copy number variations, from these SNP arrays, including Bird Suite here at the Broad and PennCNV. And these could assess Copy number variation, again, down to a somewhat smaller scale. It depends on the density of SNPs on the arrays. Initially, there were hundreds of thousands of SNPs, and then eventually millions of SNPs. And with a high-density array, you can get down to something like tens of kilobases, maybe 50 kilobases and up, and you can detect copy number variation. You can detect deletions, and you can detect duplications, but maybe not really extreme copy number changes. And then probably the next phase in the evolution is that sequencing data came along, and so we had short-read sequencing. And there's several different ways you can use short-read sequencing to try to assess copy number variation. It's kind of a challenging problem because you're generally looking for a large structural change using a relatively short read. But there are signals you can use. So for example, most sequencing today is paired-end sequencing. So if there's no structural variation, you know about how far apart each pair of reads should align when you align it back to the reference genome. But if there was a deletion, for example, then what would happen is those reads would actually align farther apart than you would expect because the DNA you sequenced was actually shorter than the reference genome. And similarly, you'd get, for an insertion of novel sequence, you'd see an unpaired read where its mate was sequenced you'd just never seen before in a human genome. And for tandem duplications, you would get read pairs where the orientations looked wrong because there was actually extra sequence in the reference that was in the sample that was kind of unexpected or different from the reference. You can also use things kind of in the same sort of idea that you could use the fluorescence intensity to assess the DNA abundance. You could just count how many reads you see at each location in the genome. And there's some stochastic variation, but you can count the reads and get some idea whether or not there's more DNA present or less. If the DNA is missing completely, like in this example in the purple, you will in theory see no reads there unless there's mismapping. So you can detect duplications and deletions this way. And if you have long enough reads, you can also look for reads that actually, where part of the read aligns one place in the genome and part aligns someplace else, and we usually call this split reads. And as the read lengths get longer, you get more and more power to detect structural variation in this way. So with the advent of these sequencing technologies, then there were methods developed to utilize the sequencing technologies to call structural variation. And if you have targeted exome sequencing, where you're doing exome capture, you can look at, usually people mostly look at the depth of coverage signal in each target exon that's been sequenced. And there's a certain amount of noise in this introduced by the capture, but if you normalize out the noise, you can call copy number variation. It depends a little bit on the algorithm. Usually you need 2 or 3 exons in a row with a copy number change. And you can usually just tell deletion and duplication. Some of the early work in this was from, for example, Menachem-Fromer, who wrote XHMM, and then GATK4 now has an exome CNV caller that works pretty much on this same sort of idea. So along with exome sequencing, of course, we began to have whole genome sequencing, and there you can ascertain structural variation, outside of just the coding regions. And as opposed to exome sequencing, where the breakpoints are maybe unlikely to land right in an exon, if you've got the whole genome, you can also use the breakpoints a lot. So several methods have been developed to try to call structural variation from whole genome sequencing. The one developed in our lab is called genome strip, and it was developed in the context of the 1,000 Genomes Project. And in benchmarking in the 1,000 Genomes Project, it was generally, and I think always, the most specific algorithm when we benchmarked it, and it was usually at or very close to the top in terms of sensitivity. And a couple of the things we learned in that experience were very important. One is that genome strip is a population-based caller. So it works on at least tens of genomes, ideally hundreds of genomes from different people pooled together. And in doing this, this actually corrects for a lot of technical artifacts and makes the problem quite a bit easier if you have the abundance of data to do that. And the other thing that was learned in this time frame is that of these 3 signals, the read depth and the split reads and the read pairs, any algorithm that combines at least 2 of these signals does much better than an algorithm that's using just 1 of them. And this is because there's sort of different error modes in each of the signals, and if you're getting corroborating evidence from at least 2 signals, this improves your calling accuracy quite a bit. And most of the good calling algorithms today are using multiple signals together in this way. So another sort of sub-problem of structural variation is that there are these mobile elements, these retrotransposons that exist in the human genome. So these are DNA sequences that can cut and paste, or in the ones active in humans, they copy and paste. So the retrotransposon can create a copy of itself somewhere else in the genome, and then that copy can persist over time and even create additional copies. There's three major kinds active in humans. There's alleles, which are about 300 base pairs. There's L1s, which when they're full length are about 6 kb. And it's important because L1s have to be full length to then duplicate again. And then there's the third kind is SVAs, which are a little less common than the alleles and L1s. And there have been some methods developed specifically to target these mobile element insertions, because these will show up in different people and be polymorphic in the population, but they're not in the reference, so they're a little hard to detect. Some of the initial work on this was done by Chip Stewart when he was at Boston College, and now he's a Brody. And probably the most popular software today for doing this that I know of anyway is MELT from the University of Maryland, and from benchmarks I've seen in the 1,000 genomes and other places, it seems to work pretty well. And last, I want to talk a little bit, and probably not nearly in the depth it deserves, about work that's been done, especially in Mike Talkowski's lab, looking at long insert libraries. So the idea here is that you prepare a sequencing library where the DNA fragments are really long. Mike told me like 3.5 kb is pretty typical. I think if you want to push the envelope, you can get up to 6 or 7 kb. So the idea is when you have these really long reads, you get really deep, what we call physical coverage of the genome. So you have a certain amount of sequencing, but the total physical distance you're spanning with all those molecules is really, really high. And if you use the information that you've got these long molecules, and then you see the read pairs moving around where you don't expect it, you can detect large variations, like large deletions and duplications, and especially inversions and translocations. And the Talkowski lab has used this in a lot of very good work that you can look up online, especially in more rare diseases and rare disorders where this kind of variation, these translocations and inversions and things are more frequent and probably more important. So if you kind of put this all together and look back at kind of the 2011, 2012 timeframe, here's one of the results from 1,000 Genomes Phase 1 in the structural variation working group. And using all of this, this short read sequencing data at low coverage. So the 1,000 genomes was at this time about 1,000 people, and it was 4 to 6x sequencing coverage. And this was the spectrum of structural variation that was discovered using the methods that were available at the time in that data, and using multiple different methods, like 10, 12, 15 different methods, and pooling and really QCing all the calls that came from those methods. And you can see a couple things here. One is that as the size of the structural variation gets larger, they get less and less frequent. And as you go to a shorter length scale, and the length scale here is on a log scale, as you go shorter and shorter, they get more frequent. You see this really big spike, which is at that 300 base pair size range, and these are alleles that are polymorphic in the human population. And then you see this increase, and then you see it sort of tail off toward the end as you get below, down here, below 100 or 200 base pairs. And that's not really because variation doesn't exist at that scale. It's because that in that data set, there was just no power to ascertain that size of structural variation. It's a size range that's particularly refractive to this short read technology. Small SNPs and indels fit within a read. Really large structural variations, you have many reads you can interrogate to see the signature of the structural variation. But when you have something that's just a couple 300 base pairs, you don't have many reads that are informative about that variation. So it's a difficult area for short read technologies to access. So what would make the situation better, right? So what would make it better would be long reads. In fact, the holy grail of structural variation calling would be really long reads, really high fidelity, base pair level, no errors, and add in high throughput and cheap, and then we'd be all set, right? We could interrogate all the variation in the human genome, and then we'd just be down to biology. So one technology we have available today is PacBio sequencing. And PacBio sequencing has fairly long reads, 10, 20 kb, even somewhat longer. And you can use these to resolve structural variation. The downside is there's a pretty high error rate. With PacBio, the base error rate and the indel error rate is pretty high. So you need fairly deep sequencing to be able to error correct all of the errors that are kind of inherent in the PacBio sequencing technology. Nonetheless, in the last phases of the 1,000 genomes, we had PacBio data on some number of individuals, and one of the results from that was focused on is that we cataloged a certain amount of this kind of complex variation that exists not in people with disease, but in just the healthy human population. So these are inversions or sort of complicated inversions and rearrangements that seem to be segregating in the population at different frequencies. The numbers of these are, as you can see here, in the like 100, 200 range, having surveyed 2,500 people. So they're not incredibly prevalent, but they do exist in the healthy human population. And this PacBio technology has also been used in a slightly different way, which is to create very high quality personal genomes. So we take a single genome and we sequence it very deeply with PacBio. Maybe we use other technologies. We combine that with short reads, and we essentially get a brand new human assembly of a single person. And then as we accumulate these genomes over time, we have an excellent reference for the range of structural variation that exists in the population, at least the common variation. So far, this has been done on kind of tens of genomes. There is the Genomes in a Bottle Consortium that's done the Ashkenazi trio. There's ongoing work in the 1,000 Genomes Consortium where 3 trios are being done, 1 Chinese, 1 Puerto Rican, and 1 Yoruban. And there's a few more genomes like this. But due to cost, the number of samples that we've been able to do this for so far has been fairly low. But one of the samples that has been done is this hydatiform mole sample. And for those of you who don't know what this is, and I'm not a biologist, so you have to bear with me here, but my understanding is that an egg can be fertilized, an egg cell can be fertilized, and also once in a while it will lose the maternal DNA. And then what happens is during the first replication cycle, the DNA from the sperm replicates, and so the cell becomes diploid, but it's got 2 copies of the paternal DNA and no maternal DNA. So that cell is effectively haploid. It's not truly haploid because it has 2 copies of the DNA, but vis-a-vis mutations that happen after that first cell division, those 2 copies are identical. And this cell isn't viable. It can't turn into an embryo, but you can make a cell line from it. And this has been done at least twice. There's one called CHM1 and one called CHM13. And these are really excellent reference samples because they're haploid. It's very easy to call variation because everything's homozygous. And so this is some data from work that was done to deeply sequence and assemble one of these haploid genomes. And this probably gives us the best picture of the full spectrum of structural variation because it was done with PacBio sequencing and short read sequencing as well. And one of the things you can see here is that the length spectrum is being shown here on the left panel. It's showing the length spectrum of variance under 1 kb, and on the right panel, to sort of scale it up, it's variance over 1 kb. And I'm showing over here the 1,000 genomes for comparison. It's a little awkward because this is on a log scale, but you can see that there's kind of this general sort of exponential trend of the number of variants, in this case, starting with 50 base pairs and up, so that as you get longer, there's fewer and fewer variants with these sort of typical peaks at the size range of ALUs and over here at the size range of full-length L1s. And this trend basically also continues, as far as we know, as you go from 50 base pairs all the way down to small indels and SNPs. There's kind of this exponential decay in the number of variants. So part of the message, the first part of this talk is very focused on technology, and that's partly because what we know about structural variation has been informed a lot by the technologies we've had available to interrogate structural variation, right? As we develop new technologies, we can learn about structural variation more, and especially in places that have been refractory before. So I want to talk a little bit more about 2 other technologies briefly. One is optical mapping, and a version of this has existed since the 1990s that was based on stretching out DNA and using restriction enzymes to cut it and then measuring the fragment lengths. In the last few years, BioNano has a similar technology, but it's based on using a nicking enzyme, and then during the repair process, you fluorescently label the nicks, and you stretch out the DNA, and you can measure the nicks. And so you get sort of this pattern of nicks, and you can use that pattern of nicks to reassemble the molecules into a picture of what the original structure was. The real advantage of this technology is you get nice long molecules. You can easily get molecules 150, 200 kb and up. So if you've got a very long structural variation or something very complex, you have a lot of power to span across it and see the full range of structure. The downside, of course, is that you only see where the nicks are, and you have no idea what the sequence is that's in between those nicks. You can try to infer it and put this together with other data. But this is a really powerful technique if you really need those long molecules. Another technology that's fairly new in just the last year or two is these linked reads from a company called 10x Genomics. And the way these work is that they've got this microfluidic technology where you take nanoliter-sized droplets and you have these little particles that they call gems that I believe actually dissolve. And the trick is you trap a DNA molecule and one of these particles in the same droplet. The particle dissolves, it's got barcoded primers in it, and all the primers have the same barcode. They kind of do their thing, and you make a library of regular Illumina sequencing, and then you can pool the results of all these microdroplets together. And the key is that the molecules you put in, you want to be nice and long, typically like 40 kb or something. And because all those primers in the gem had the same barcode in them, when the sequencing reads come out, you've got this barcode that tells you which long molecule the reads came from. So you've got a bunch of Illumina paired end reads, and you've got this barcode that gives you long-range information. You typically sequence at very low depth, so each of those 40 kb molecules only has like 0.1x coverage. But you do many, many molecules, so in total you have 20 or 25x coverage, like you would for a deep whole genome. But you've also got this long-range barcode information. Well, that's great, but what can you do with this long-range barcode information? Well, there's at least 3 different ways that people are exploiting and trying to exploit this long-range barcode information. The first way is you could just use those barcodes to try to detect structural variation directly. And there's this long-ranger software from 10x Genomics that takes this approach. And the conceptual idea is because those barcoded reads all came from the same long molecule, they should be close to each other when you align them back to the reference. And so when the genome is normal and there's no structural variation, this heat map is showing you on the left that all the barcode buddies for each molecule are nearby. They're nearby with about the length of the molecules that went into your reaction. But if there was structural variation, then what happens is some of those barcodes are not near their buddies because the genome you sequence doesn't look like the reference. And so you get different patterns. On the right, this is the kind of pattern you get if there was an inversion. In this case, a homozygous inversion, so the pattern is really clear. The heat map is showing that the buddies are not near each other when you align them back to the genome. If you took the reference and flipped that segment, though, all the buddies would go back to being near their friends. So you can use this to call structural variation, and that's one approach people are using with this long-range information in the linked read barcodes. So a second way that people are exploiting this linked read barcode information, I think is very clever. And the idea is this. You take these linked reads, and the first thing you do is call SNPs, just kind of regular SNP calling, and you look for heterozygous SNPs. And once you've identified a bunch of heterozygous SNPs, you try to phase the reads. You say which reads are contributing to the A allele or to the C allele at any given location. And the reads have barcodes. So by phasing the reads, you're phasing the barcodes, which phases all the reads that come from those buddies. So you use the phasing of those SNPs to partition your reads into 3 piles. Reads that come from haplotype 1, reads that come from haplotype 2, and reads where you couldn't get a consensus phasing from them, perhaps because there just weren't enough reads on that long molecule that happened to hit heterozygous SNPs. And in general, I'd say maybe you get 1/3 of the reads in each of those 3 piles. But once you've partitioned the reads by phasing, now you can go back and do structural variation calling in each partition separately. And what this does is it changes the problem of detecting a diploid variant into the problem, a heterozygous variant, into detecting a homozygous variant, but just with fewer reads. But this is actually a great gain in power. So for example here, this is showing a very small 800 base pair deletion, and this is showing just the signal you would get from read depth of coverage. This would be very difficult to detect in a standard 25x Illumina genome. You would have so much statistical fluctuation in the coverage, it would be very hard to detect this heterozygous deletion. But you can see here on the blue reads from haplotype 2, there's almost no reads. So it's pretty clear that you can detect that deletion. So this is sort of an illustration of the power you gain by first phasing the reads and then using the phasing information in your calling. And you can apply that to all the structural variation calling in all sorts of ways, looking at the breakpoints and the split reads and everything.
Just a question, by phasing there, you mean phasing within a haplotype, not looking to the parent of origin. I mean, we think of phasing almost always in genetic terms of looking for parental DNAs and which word it comes from. You're talking about just phasing it as to a haplotype.
Just as to which haplotype, and it's even just local, right? Within this region, I had two haplotypes and very far apart. Maybe I don't know if the haplotypes crossed or not, but locally, I know I can partition my reads into two buckets. I know they go approximately here, and half are on this haplotype and half are on that haplotype.
Can you then use that to phase things from parental reads without the parents? In other words, look at all those which are phased and see if they're on the same haplotype. I'm using phasing in two different directions.
Yeah, I mean, you'd have to have some information about the parent to know parent of origin.
So, a lot of trios, we only have one parent. Would this help you then phase it?
I guess I'd have to think about it, but it seems...
This is important for de novos. When you're trying to think about detecting rise de novo, you have a trio, you only have one parent, and the mother doesn't have it, the father have it, or is it de novo? Can you put enough data together to be able to do that? And maybe this is a little orthogonal to what you're doing. You may want to take it offline.
Yeah, I'd have to think about it, whether or not you could really rule out, whether you could really prove that de novo was from the one parent you have. I have a feeling you can't, but I'd have to think about it. So let's take it offline so I don't mess up. So I wanted to also talk about the third thing that you can do with this long-range information, which is that David Jaffe, who was formerly a Brody and worked on many assembly algorithms here, including most recently Discovar, has gone to 10x and is working there in conjunction with other people on a new assembler called Supernova. And preliminary results suggest that using this long-range information from the linked reads, it's enough to do pretty decent assemblies just from the linked reads. You don't need long insert libraries, you don't need PacBio, but you can get pretty good personal genome assemblies just from the linked reads and enough computational power. And this is important because it really drives down the cost of doing these personal genomes. And if you can do even a pretty good assembly of these personal genomes, this could be, I think, potentially, if the assembly can work as well as we might all hope, that you could do a very good job exploiting that to look at structural variation. So that was a lot of different technologies and how we might be able to exploit them to look at structural variation. So I want to sort of switch gears and talk a little bit about the genetics of structural variation and what we've learned to date about the properties of structural variants, at least some of them, and things that at a primer level is probably good to know, in my opinion. So the first thing we've already kind of talked about, and I want to go back to it again, and that's this idea that there's kind of this exponential decay in the number of variants based on variant size, with the exception of these retrotransposable elements in human genomes. And sort of traditionally, I guess, in the structural variation field, there's been kind of this way of partitioning the problem in terms of looking at it as this menagerie of structural variation, where we try to take different types of variation that we see in humans and categorize and classify them. And this is kind of the classification we usually use. So one of the first classifications is just deletions and duplications. So you've lost some DNA sequence, you've gained some DNA sequence, second copy of existing sequence in the human. For most of the data we've seen in our hands, and I think most of the studies out there, I would say that in general, the numbers of deletions and duplications relative to the human reference are roughly equal. And the number, it's a little tricky to put a number on them because it depends strongly on the length scale. So anytime you look at the number of variants, deletions and duplications someone found, you always have to ask, well, what length range were they able to ascertain, right? Because of this exponential drop in the number of variants with size, if you can ascertain at 1 kb, you're 1 kb and up, you're going to get one number. As soon as you can ascertain down to 700 or 800 base pairs, you're going to get a much higher number because there's many more variants as you go to smaller length scales. But in general, you know, there's tens, maybe hundreds of thousands of deletions and duplications at reasonable frequency in the population. Where the exact number sort of depends on size, but it gives you sort of a rough feel of that part of the menagerie. There's also inversions and translocations and these kind of complex rearrangements I talked about before. These tend to be more in the hundreds, like I said before, although they do segregate in the human population. One of the things that's been learned, and there was a lot on this in 1,000 Genomes Phase 3 and other papers, Mike Telkowski's lab has shown this a lot. There used to be this idea that there would be a lot of inversions that were just kind of your classic flip the sequence end for end. But it turns out that an awful lot of the time, when you see an inversion, it's coupled with something else. The sequence inverts, but maybe it duplicates a little bit farther away. That's kind of what this dot plot here is showing. And this is aligning, these dot plots are aligning long reads back to the reference genome, so you can kind of interpret them by how the DNA moved around. And sometimes, as these arrows are showing, there's a lot more complicated patterns of inversions and rearrangements that went on. We talked a little bit about mobile elements. So and probably one of the things to understand about mobile elements and sort of structural variation detection and measurement is that the mechanism, the biological mechanism for mobile elements is a copy and paste mechanism. So biologically, the mobile element is always an insertion relative to the genome that was in the ancestor. But a certain amount of these mobile elements are captured on the human reference. So when you compare your data to the human reference, you might see a mobile element that appears to be deleted. And that's just because the reference has the insertion haplotype. And the DNA you sequenced has the non-inserted haplotype, so it appears as a deletion. And you can also see mobile elements that appear to be inserted into the reference, and that's just because the human reference doesn't have them. So our perception of them and the way you measure them is sort of polarized when we use the human reference. I think I sort of mentioned this before. ALUs are the most frequent in terms of what's an active element in humans, and then L1s and then SVAs are a little bit less common. So I don't have a lot to say about novel sequence insertions because I haven't looked at them too much. A little bit, it's definitional as to whether or not, you'll see reports of, we found this many megabytes of novel human sequence. It's a little bit definitional in terms of whether that novel sequence is an awful lot like sequence that's already in a human genome, but just a little bit diverged, or whether it's really kind of very different new sequence that's human that we've just never cataloged before. I haven't looked deeply at the data here. My impression is that the truly novel human sequence, we don't see that much of it, that sort of our catalog of the human reference, and if you include all the unplaced contigs and stuff, is actually fairly complete, except for maybe polymorphic duplications of existing human sequence. But I haven't looked at this in great detail. I am going to spend a little bit more time talking about multi-allelic CNVs, mostly because this is near and dear to my heart. It's a lot of what our lab works on, and it's a lot of what my research has been focused on in the last couple years. Oh, there we go. So traditionally, people thought of deletions and duplications as kind of existing in isolation, right? That there would be like this one region of the genome and maybe it's lost in some people, and occasionally you would get a segment of DNA and you end up with 2 copies of it on one haplotype, and that these would be kind of distinct events that would segregate in the human population, kind of like SNPs do. But actually, when we began to look, there were some loci that behaved like that, but there were also a lot of loci where it just seemed like something much more funky was going on. There's lots of copy number variation. It's all in the same region of the genome. There's different segments varying, but they're not correlated with each other. And it's we've come to realize that there's places in the genome where there's a lot of complex structural variation that goes on. Not everywhere, but certain loci seem to be especially like this. And so some of the work that's been done in Steve's lab has looked at these multi-allelic CNV loci. We had a manuscript on this a couple years ago in Nature Genetics. And this is kind of what these multi-allelic CNV loci look like when we analyze them. But what these histograms are showing here is the genomes of many, many people, and we're classifying them according to our estimated copy number, diploid copy number, so the sum of the number of copies of this DNA segment on both alleles. And the colors are showing you the integer copy number we're assigning. So if somebody's homozygous for the reference, they're going to have a diploid copy number of 2, and they'd be in blue on these charts. And you can see here that at these particular loci, there's just a wide spectrum in the number of diploid copies that different people have across the human population. So these are obligately multi-allelic because there's no way 2 alleles can explain this range of variation. And it's also pretty obvious that these have to be highly polymorphic sites in the population. And one more technology I'm going to talk about briefly is droplet digital PCR, something that we've used heavily in our lab as a way to measure copy number variation. And we especially used it a lot in this multi-allelic CNV analysis because we wanted to know if our inferences from sequencing data, which is what I was showing on the previous slide, are accurate and well calibrated. And the way ddPCR uses these nanoliter-sized droplets. And the idea is you take your DNA sample and you dilute it down so that when you put it into these droplets, you try to get exactly 1 molecule per droplet. So most of the droplets will have 1 molecule, some will have none, a few might have 2, and then you do what they told me is sort of a regular TACMAN assay inside the droplet. So you have a control locus and you have your DNA, and then it either fluoresces like red or green. But then if you've done a good job and you only have 1 molecule per droplet, then the droplet is either all red or all green, or no color if you didn't get any molecule in it, or it might, if you got 2 molecules, it might be red and green. But once you've done that, now you just count the droplets. How many red droplets did you see? How many green? And it tells you the copy number of your target locus relative to your 2-copy control locus. So this is kind of like qPCR, but much more accurate because you've got this digital counting going on in the droplets. And this is really the only cost-effective way we have to measure copy number at these kind of multi-allelic CNV regions with a wet lab technique. Droplet digital PCR, you can, if you do a few replications, get the copy number at copy numbers of like 10, 11, and 12 even, accurate to integer values. And we were able to show that our sequencing analysis and the ddPCR agreed with extremely high concordance, and this gave us a lot of confidence in our sequencing measurements. So what did we learn about these multi-allelic CNVs? Well, one thing we looked at was just the abundance of them. How many are there? On the top row here, we're showing in color just biallelic deletions in blue and biallelic duplications in orange, and then the multi-allelic CNVs in green. And we found that there were less of these multi-allelic CNVs. There's more deletions and duplications in what we were able to ascertain here using the 1,000 genomes data. But when you looked at just CNVs that overlap genes, we found that the distribution shifted. Right? In the second line, when you look at genic CNVs, the number of deletions drops dramatically. The proportion of deletions drops relative to the duplications and multi-allelic CNVs. And this suggests that a lot of genes just can't tolerate haploinsufficiency, or not enough to allow there to be common deletions of those genes segregating in the population. And that duplications and these multi-allelic CNVs are more tolerated in general. And when you get these deletions and duplications of the genes, we look at this as kind of a gene dosage change, right? So how many copies of gene X does this individual carry? Do they carry 2 copies? Do they carry 4 copies? Do they carry 5 copies? And then we asked about differences in gene dosage variation. We said if we take all the genes that appear to vary in dosage among people, and you ask how many times do they vary between 2 individuals, like any 2 individuals you would pick in this room, well, how many of us have different numbers of copies of some genes? And the answer turned out to be about 75. So you pick two people in this room, probably, and you count how many copies of gene A do I have, how many copies of gene B, how many copies of gene C. You probably differ in copy number in 75 genes. And this is skewed tremendously. This is driven almost entirely by these multi-allelic CNV loci. These are contributing overwhelmingly to the amount of gene dosage variation that's happening between any two people. And this was initially a somewhat surprising finding, the strength of this effect, that it was so pooled in these multi-allelic CNVs. And then we dug down into this by looking at the frequency of the variation. What we found is if you look at low frequency variation here in this top circle, looking at variation that's below a 5% frequency, we found there's kind of this balance between deletions and duplications and then the multi-allelic CNVs shown in green. But then when you look at common variants, it changes. There's still a reasonable number of deletions, but the duplications almost disappear, right? There are almost no high-frequency biallelic duplications that we see in humans. So this leads to the following idea, is that what's going on is that perhaps duplications are just not a very stable state of the genome evolutionarily, a biallelic duplication. Because once you've got a duplication allele, you've got two copies of a DNA sequence, now you have the possibility to do unequal crossover during meiosis, right? So over time what happens is you'll get an unequal crossover and you'll suddenly get a 3-copy allele. And maybe that 3-copy allele will revert back to a 2-copy allele. Maybe the 2 goes back to a 1. And you get sort of this expansion and contraction process that starts. That means that these biallelic duplications just don't persist in the population. This can't happen with deletions. So deletions can go to high frequency just based on the forces of drift and selection. But there's kind of this tendency, this natural tendency for duplications to become multi-allelic over time. So the next thing I wanted to talk about in terms of like qualitative genetics of structural variation is going back to this idea that as the variants get larger, there's fewer and fewer of them. But this is to some extent balanced by a different effect, which is that the phenotypic impact of each variant looks like it's larger for larger variants. And in some ways, maybe this is to be expected, right? We know that really large CNVs often lead to rare developmental disorders and well-known syndromes, right? So those large CNVs clearly have large phenotypic effect, right? Variant for variant. But this seems to happen more or less across the length scale. And here's some of the evidence for that. One of the things we looked at for these multi-allelic CNVs is what's their effect on gene expression. So I told you already that the gene dosage varies a lot between people. There's certain genes where people have very different numbers of copies. Well, does that actually affect the expression of the genes? In plants, there are mechanisms where if a plant acquires an extra copy of a gene, the plant can just shut it off. And this is maybe an evolutionary advantage because you don't want anybody messing with your gene dosage, right? So you want to kind of have stability. But it seems like there isn't a mechanism like this in humans. In almost every gene where we had enough data to look at it, we found a positive correlation between gene dosage and gene expression as measured from RNA sequencing. And this is showing some of the data here. So these CNVs, when they overlap genes, they have a phenotypic effect, at least at the expression level. In the 1,000 Genomes Phase 3, there was another look taken at this, also using RNA sequencing to get expression data. And then we did an analysis of eQTLs. And we asked for all the eQTLs we could find in the GeoVetus data, which use lymphoblastoid cell lines. We asked, were those eQTLs more likely to be driven by a SNP, more likely to be driven by some sort of structural variation, or there were two candidates and it was hard to tell? So this is looking at both the strength of the association and just whether or not there's a structural variation in the neighborhood. And what was found there is that in the 1,000 genomes was that SVs were probably something like 50-fold enriched for eQTLs over SNPs. They're much more rare, but sort of variant for variant, they're more likely to contribute to phenotype. And then more recently, there's a paper in review by Colby Chang and Ira Hall's lab where they looked at the GTEx data. And they did a similar analysis, but they had a lot more data. They had multiple tissue types. They had expression, so they can look at expression across multiple tissue types. And they had deep sequencing, so they were able to ascertain many more structural variations than we had in 1,000 genomes. And they found an even stronger enrichment. They're reporting that structural variations count for between 3 and 7% of EQTLs. The range is because they measured several different ways, and this was kind of the range of estimates they got for different ways of estimating. So 3 to 7% of EQTLs, despite the SVs being, despite like the SNPs and indels, there's 1,000 times more of them. But yet 3 to 7% of the EQTLs are likely to be driven by the structural variants. So another question we want to ask here is how well we can impute structural variants from SNPs. Because often if we want to see phenotypic effect, we have to get to really large sample sizes. And if we don't have whole genome sequencing on everybody, we can't necessarily ascertain these structural variants in all of the people we need to get association to phenotype. And for those who want a very quick refresher, the idea behind imputation is that there's correlation between markers that are co-located near each other on the genome due to recombination and recombination hotspots. And so even if you don't know the genotype of a particular marker, if you have a reference panel where you've got that marker and associated SNPs, you can statistically guess what that marker value is in a whole new sample where you have just the flanking SNPs but not the marker itself. And you can use this for structural variations. For example, if you have SNP arrays on a large cohort, you can impute from whole genome sequencing where you have the SNPs and the structural variation into the cohort with the SNP arrays. So we did some studies of this, looking especially at these multi-allelic CNVs and also the biallelic deletions and duplications. And we did a bunch of leave-out trials to assess, using the 1,000 genomes data again, how well we can impute structural variations from the SNPs that are next to them. And by and large, this, and actually early studies done by Steve McCarroll, showed that biallelic deletions, by and large, impute about as well as SNPs. They act like biallelic variants. If you know the SNPs flanking them, you can impute them about as well as you can impute a SNP. Your ability to impute depends strongly on the frequency of the variant. The more common the variant, the easier it is to impute. So the range of success in imputation depends a lot on the frequency of the variant you're trying to impute. So similarly, I would say that roughly duplications do as well as SNPs also, biallelic duplications. But remember, biallelic duplications are almost always rare, right? So they don't impute very well, but maybe that's just because they're rare. And rare variants don't impute as well as common variants. When we look at the multi-allelic CNVs, it's kind of a mixed bag. Some impute fairly well, some don't impute very well at all. And these plots on the right are kind of showing that this seems to be associated with things like how multi-allelic they are. So maybe how many recurrent alleles there are, what the mutation rate might be at that genomic locus. So for example, if a biallelic duplication is mostly still biallelic, maybe there's a couple extra alleles, maybe it still imputes fairly well because it's still kind of a biallelic locus. That's kind of the idea. So sort of a mixed bag on imputing multi-allelic CNVs. I'm going to try to just illustrate that briefly. I've got to maybe speed up a little bit because I'm running out of time here. But I'm going to try to illustrate that briefly with just one vignette. And this is a locus where there is this multi-allelic CNV at this gene called HPR. And you can see on the right, these traces are showing kind of the copy number classes among groups of different samples, and the distribution is shown at the top. So there is a large deletion that spans HPR that's mostly exclusive to Asians, and this deletion imputes fairly well. But there's also this multi-allelic CNV that actually had this very interesting property that it's kind of what we call a runaway copy number variant. So as opposed to the plots I was showing you before where the distribution of copy number in the population is kind of smooth, that's not true at HPR. There's a lot of people who are copy number 2, but there's very few people who are copy number 3. In fact, if you're not 2, the most likely copy number you're going to have is 6, unless you're Asian and you might have the deletion. But if you have kind of that multi-allelic locus that's over here on the right, if you're not a 2, you're most likely to be a 6. And that's pretty much seems to be an African-specific CNV. So what happens when you try to impute this? Well, the deletion imputes fairly well, but that multi-allelic CNV has the following properties. So this is something we call a spider plot. And this is showing sort of the haplotype structure around the CNV. You have to imagine that that white space in the middle is where the location of the CNV, and then out to the right and left, these are the SNP haplotypes as you walk out. This is showing the haplotypes for many, many people on the y-axis, and this blue banding is showing you the frequency of the different SNPs as you walk away from the CNV on the left and the right. And these long branches are showing you the major haplotypes that exist in the population. And here I'm showing you Africans because these are the ones that have the high-frequency CNV. And then these black tick marks here are showing you the high copy alleles, the ones that are not copy number 2 in the population. And you can see that almost all the haplotypes of the high copy alleles are on this very long haplotype at the top. And this is telling you a couple things. First of all, it's telling you that this is a fairly recent haplotype that these high copy alleles have arisen on. There's a few of them on other haplotypes, but this could simply be recombination with this long haplotype where it arose. And so if you go to impute this locus, right, if you want to ask, is somebody have the 1 copy allele or a high copy allele, that's pretty easy to impute, because you're really just asking, do they have this long haplotype or one of the other ones? But if you want to ask which high copy allele do they have between the different ones they could have up here, that's actually going to be very difficult to impute, because everybody's on that long haplotype. This data is suggesting that, in fact, this CNV is mutating at a rate that's faster than the background SNP mutation rate in its genomic locus. So that gives you a flavor of what you run into when you try to impute these multi-allelic CNVs. So I want to move on to disease impact here. So we know that there are these common disorders that are caused by large recurrent CNVs, but what about common disease? Well, one of the early examples of this was actually work done by Steve McCarroll when he was a postdoc. He found using array data this 20 kilobase deletion that was upstream of the IRGM gene. He showed that it was impacting the expression, and we knew that IRGM was associated with Crohn's disease. And so this was an early example of finding a CNV, in this case a deletion, that was having likely an impact on a common disease. Around 2007, there was some seminal work done by Jonathan Sabat, where he showed that there was this enrichment of large, rare CNVs in autism. And even though he had very few samples, he picked up this signal. And these are rare CNVs, so they're not the same CNV in everybody. Sometimes they're at the same locus, but it's also just that if you have autism, you're more likely to have these large, rare CNVs than other people in the population, all over your genome, or at least more of them, and they will be scattered around your genome. So after Jonathan reported this, many people began to look for this, and a number of recurrent loci began to emerge. So these are loci where we see that these rare CNVs happen over and over in different people, although they're different CNVs, generally occurring de novo in each subject. And so there are some common loci where these CNVs tend to show up over and over, and yet there is also evidence for a burden of just CNVs scattered around the genome. And we also see that this is true in schizophrenia, with often different loci and some shared. And probably the most recent study on this was a large study done in the PGC with Daniel Hauergen and others looking at the CNVs in particular. So a huge meta-analysis, 20,000 cases and controls across many studies, showed an odds ratio of about 1.1 for increased burden of these large, rare CNVs in schizophrenia. All right, so what about more complex CNVs, like these multi-allelic CNVs? Well, I want to share, in the few minutes I have left, a few vignettes about loci that where we've looked at these complex CNVs and association to phenotype. One of the early examples was we looked at chromosome 17, where it's known that there's this megabase scale inversion and a bunch of copy number variable segments, 3 copy number variable segments actually, that were uncorrelated with each other. And Linda Betker did a lot of work to identify at least 9 different haplotypes segregating in the human population, and found very interestingly that there was 2 haplotypes. So this H1 and H2 here is showing the 2 inversion states. And there were 2 haplotypes, one on each inversion state, which means they don't recombine with each other, that that are almost exclusively found in Europeans and have gone to quite high frequency, and both of them actually create a fusion gene involving cancel-1. I don't think this has been definitively linked to phenotype yet, but it strongly suggests that there was some sort of selection going on at this locus, although we don't know exactly why. Also, there's the amylase locus, which is incredibly polymorphic. People can have up to 20, even maybe 21, 22 copies of the amylase 1 gene. Christina Usher did a lot of work on untangling this locus, and there had been a report that this was associated with BMI, but we carefully analyzed it with a bunch of collaborators, and we were unable to replicate this association to BMI, I think mostly because we had more accurate assessment of the copy number and the structural states. An interesting thing about this locus is if you look at the copy number distribution of amylase, it sort of jumps by 2. Right? You're more likely to have 7 copies or 9 copies or 11 copies and not 8 or 10. And the reason is because when it duplicates and deletes, it tends to take 2 copies of amylase with it. Not always, but more often than not.
Maybe 1 minute, 2 minutes?
Yeah. So very briefly, Linda Becker and Ronnie Salem earlier this year published a study where they looked at the haptoglobin locus. They showed there was many complex forms of it, and show that there seems to be association between haptoglobin and cholesterol level. And I want to spend my last 1 or 2 minutes especially talking about, I can't really do it justice here, but the work by Handsaker to show the role of the C4 gene in schizophrenia. The GWAS here is showing that the strongest signal for schizophrenia among the 108 genome-wide significant loci is at the MHC. It was the first signal discovered by GWAS because it's so strong, but nobody knew what the fine mapping was for the causal area behind this. Ashwin showed that at the C4 gene, there's these complicated structures, and that in fact that signal looks to be almost exclusively driven by these structures at the C4 gene. The C4 gene exists in 2 different forms, A and B, that differ by 4 residues at one particular spot in the gene. And there's a HERV that inserts into intron 9, which creates both a long and a short form. And then in addition to this, it's copy number variable. If you look at enough people, you'll see pretty much the whole spectrum of the number of C4a and C4b copies and the long and short forms segregating in different people. And the final important point here I want to make is that Ashwin showed that the association was to the C4 copy number and not to the SNP haplotype. Before I told you about how it can be difficult to impute these CNVs because they exist on different SNP backgrounds, well, that's both a blessing and a curse, right? It makes it difficult to impute them, but when you can impute them, because the CNV is existing on multiple SNP haplotypes, that gives you the power to test whether the association is to the copy number state or to the SNP haplotype. And Ashwin showed that the effect here, it seems to be associated with the copy number structure and independent of which SNP haplotype it's on. So a summary of what I talked about, there's fewer SVs than SNPs, but there's more impact per variant. In some cases, it looks like they have faster mutation rates. They're not always well tagged by SNPs, but that's kind of a mixed bag. And there's a lot of work to do on more analysis of structural variation. And thanks to all the people who gave me slides and collaborators and to Steve McCarroll, who runs the lab. Thank you all.